Hey everyone! In the last video, you learned about the cellular and the genetic differences between prokaryotes and eukaryotes. Now in this video, you'll learn how prokaryotes carry out cell division. And by the end, you should be able to answer one question. And our question is, how does binary fission occur? All cells must accomplish three things in order to produce a copy of themselves. They must first make copies of their chromosome or chromosomes. Second, they must separate those copies. And third, they must divide the cell in such a way that the two new cells, called daughter cells, get the same number of chromosomes. Prokaryotes accomplish these three things through a process called binary fission. This is a form of asexual reproduction because only one organism, in this case a single-celled organism, is required to reproduce. Before binary fission proceeds, the bacterial chromosome exists in a coiled state. During fission, the chromosome is uncoiled. The chromosome and any plasmids are then replicated by the process of DNA replication. The original and newly made chromosomes are moved towards the opposite ends of the cell, while the cell grows and becomes longer. A new cell wall then forms near the middle of the cell, separating the two cells. The chromosomes then recoil in their new cells. The chromosomes in each daughter cell, assuming no mutation occurred, are exact copies of each other. So the same alleles of the same genes are on these chromosomes. They're clones. So these cells have the same genotypes, and remember, since bacteria are haploid, their genotypes only have one allele of each gene. Single-celled eukaryotes, like our friend the euglena, can also reproduce asexually through a process similar to the binary fission we just described. In eukaryotes, some organelles, including mitochondria, because they evolved from free-living bacteria, also divide by binary fission. Replication of the mitochondria ensures that once the cell divides, each daughter cell will have enough mitochondria. Problems with mitochondria are thought to play a role in several age-associated and neurodegenerative diseases, including Parkinson's disease, Huntington's disease, and Alzheimer's. So binary fission is important in humans as well. Because binary fission is simpler than eukaryotic cell division, and because there's less DNA to be replicated, it's faster. Under ideal conditions, E. coli cells can divide about every 20 minutes. So if we start with 100 cells, after 20 minutes, or one generation, there are 200 cells. After another 20 minutes, there are 400 cells. And after about 5 hours, there will be more than 1 million cells. Something you might want to think about when you're considering whether you want to eat that potato salad that's been sitting in the sun for a while. Then again, maybe you like to live dangerously. Now that you know how binary fission proceeds and how quickly bacteria can divide, follow the link below this video to check out a time-lapse video of bacteria dividing by binary fission, and to learn why bacteria don't just take over the planet with their rapid growth rates. Okay, so that's how bacteria divide through binary fission. Eukaryotic cells, on the other hand, divide by mitosis and meiosis. But before we get to that, we'll spend some time examining eukaryotic chromosomes. See you later.